Hey everyone, ready for a deep dive? We're going to be tackling some pretty mind-bending stuff today,
Mhm.
exploring psychedelics and how they're changing the way we think about the brain. We've got five uh, key areas to explore, and they're all pretty wild. Think ego resets, the brain's secret psychedelic messengers, and even ways to control neural circuits remotely.
Yeah, it's a really exciting field for sure. There's so much potential, and we're finally starting to see a shift away from the old stigma and focusing more on the science.
Okay, so let's dive right into that first one, the ego reset. We all get stuck in those negative thought loops.
Mhm.
What if there was a way to hit a reset button on those patterns?
Well, that's where psychedelics like psilocybin come in. Research is showing that they can temporarily disrupt the default mode network in the brain, or DMN. It's the network that's really active when our minds are wandering, and it plays a big role in our sense of self or ego.
So by disrupting the DMN, are psychedelics essentially silencing that inner critic, the one that's always telling us we're not good enough?
Exactly. Psilocybin seems to create a sense of openness and interconnectedness, quieting that inner voice and allowing for new perspectives to emerge. You could think of it like rebooting your mental operating system.
Okay, I'm starting to see the appeal,
hmm
but what does this actually look like in practice?
So imagine you're struggling with a creative block or feeling trapped by limiting beliefs. Psilocybin, in a controlled therapeutic setting, can help break down those rigid thought patterns and make space for new ideas and insights to flow.
It's like clearing out the mental clutter so you can see things with fresh eyes. I've even read about research using psilocybin to help people confront their fear of death.
Yeah, that's right. By diminishing the ego, psilocybin seems to ease the anxiety around death. It's fascinating research, and it really highlights just how profound these compounds can be in changing how we understand ourselves and our existence.
Okay, on to the next mind bender, DMT. I've always been fascinated by it, the whole super potent hallucinogen thing. But you mentioned something even wilder. Our brains actually produce it naturally.
It's true. We all have this powerful psychedelic molecule, dimethyltryptamine, inside us. It's still a bit of a mystery why we produce it, but we do know our bodies break it down very quickly with an enzyme called monoamine oxidase, or MAO. That quick breakdown keeps us from, constantly tripping out.
So it's like a built-in safety mechanism. But what happens if you bypass that mechanism?
Well, that's where ayahuasca comes in. It's a traditional brew that combines DMT with a plant that blocks MAO. So the DMT sticks around a lot longer, and that's what leads to those long, often profound psychedelic experiences.
Wow, that's quite the concoction. So we make DMT, but we also have a way to regulate it.
Hmm.
What are the theories about its role in our normal brain function? What's all that DMT doing in there?
It's a puzzle. Some researchers think it plays a role in dreaming, while others suggest it could be connected to near-death experiences or even consciousness itself.
DMT just went from powerful hallucinogen to mysterious brain molecule in my book. Things are getting pretty meta here. Ready to switch gears a bit?
Absolutely. Let's talk DREDs.
Okay, Dread Ds. The name sounds like something out of a sci-fi movie. What are we talking about here?
It stands for designer receptors, exclusively activated by designer drugs. They're basically engineered receptors that scientists can put into specific brain cells. Think of it like installing a custom-made lock on a door.
Okay, so we've got the special lock. What's the key?
The key is a designer drug. When you give this drug, it only activates those specific D-dreads, triggering a response in the targeted cells. It's kind of like having a remote control for specific brain circuits.
So instead of sending a general signal to the whole brain, you can really zero in on specific areas. What are the implications of that level of precision?
It's revolutionizing research. Imagine you can activate the fear center in a mouse's brain and see exactly how it behaves. Or flip it, activate the reward center, and study the pathways of pleasure.
Okay, I'm starting to see just how powerful this could be. What about therapies? Could Dread Ds be used to treat human conditions?
It's being researched actively. Scientists are looking at how DREAD-Ds could be used to treat things like Parkinson's disease, addiction, even mood disorders. The idea is to target the specific neural circuits involved and kind of tune their activity.
It almost sounds too good to be true. To be able to target those circuits so precisely, that could be a game changer. But before we get too deep into that, let's talk about a different kind of potential, psychedelics that don't cause hallucinations. Is that even a thing?
It is, and it's a really exciting area of research. These non-hallucinogenic psychedelics might offer the same therapeutic benefits without the intense psychedelic experience, which some people find really challenging. It seems like they work by enhancing neuroplasticity.
Neuroplasticity, right. That's the brain's ability to change and adapt, like rewiring itself.
Exactly. It's the foundation of learning, memory, and our ability to recover from injuries to the brain.
So these non-hallucinogenic psychedelics are basically boosting the brain's power to rewire itself. What are the potential benefits there?
Well, imagine being able to overcome thought patterns and behaviors that are really deeply ingrained, things that contribute to conditions like PTSD or depression. These compounds could potentially help people break free from those rigid pathways in the brain and create new, healthier ones.
Fascinating stuff. I have one more question before we wrap up this part of our deep dive. I've heard a lot of buzz about microdosing. What exactly is it, and what's the science behind it?
So microdosing involves taking really small, subperceptual doses of psychedelics, usually LSD or psilocybin.
Subperceptual, meaning you don't actually trip.
Exactly. The dose is so tiny you don't experience any noticeable psychedelic effects. The idea is to get these subtle improvements in cognitive function, creativity, and overall well-being.
So it's like a little brain boost.
Yeah.
Without the full-on psychedelic experience, are there any risks involved?
Microdosing is gaining popularity, but the research is still really early. It's important to approach it with caution and be aware of any potential risks.
Definitely. Is there any evidence that it actually works?
Some people report that it boosts their creativity and focus, but we need a lot more research to really understand its effects.
So it sounds like microdosing is still a bit as a mystery. We've covered a lot of ground in this first part of our deep dive. Ego resets, the enigmatic DMT, the precision of dreadies, non-hallucinogenic psychedelics, and the allure of microdosing. It's pretty clear we're just scratching the surface here.
Yeah, we've only just begun to explore how these compounds might change the way we understand the brain and open up new possibilities for treating all kinds of conditions.
My mind is already buzzing with questions and ideas. I can't wait to see what other transformative factoids we dig up in part 2.
Welcome back to our psychedelic exploration. I'm curious, has this deep dive made you rethink any of your beliefs about how our brains work?
Absolutely. It feels like we're peeling back layers of assumptions and uncovering a whole new world of possibilities. We left off talking about microdosing, but now I'm thinking about the bigger picture. What about the FDA? Where do they stand on all this research?
That's a great question. The FDA has historically been very cautious, especially when it comes to substances that have a recreational use history, but we're seeing some signs of a shift.
Really? What kind of signs? Because it seems like psychedelics still carry a lot of, well, baggage, even with all the research that's being done.
We'll just look at psilocybin. The FDA has granted it breakthrough therapy designation for treatment-resistant depression.
Breakthrough therapy, that sounds like they're acknowledging the potential there. What does that designation actually mean?
It's a big deal. It basically fast-tracks the drug development process. It means the FDA recognizes psilocybin's potential, and they're willing to expedite the review process.
So things are definitely moving forward, at least with psilocybin. What's driving this change? Is it anecdotal evidence, or is there solid science backing it up?
The scientific community is really stepping up. We're seeing really rigorous studies using brain imaging like fMRI to actually observe how these compounds affect brain activity.
It's one thing to hear about people's personal experiences, but it's another thing entirely to see the physical evidence of how psychedelics are impacting the brain. What are some of the key findings from these studies?
The data is showing us that psychedelics can actually change brain connectivity in pretty profound ways. Remember those rigid thought patterns we talked about? Well, psychedelics seem to break them down, and that allows for more flexible, adaptable thinking.
So it's like they're shaking things up, letting the brain reconfigure itself in a healthier way. But with all this talk about rewiring and potential benefits, we have to talk about the risks too, right?
Absolutely. It's crucial to remember that these are powerful substances and they're not for everyone. They can be very emotionally intense, and in some cases, they can even trigger anxiety or psychosis.
So this isn't something to just jump into lightly. What are the factors that determine whether someone will have a positive or negative experience?
Set and setting are super important. Set refers to the person's mindset going into the experience. Are they feeling emotionally stable? Do they feel supported? Setting is the physical and social environment. Is it safe and comfortable?
So it sounds like if someone is going through a tough time or they're in an unstable environment, it's probably not a good idea to be experimenting with psychedelics. Responsible use is key.
Absolutely. And having a trusted guide or facilitator, especially if it's someone's first time or if they have a history of mental health challenges.
That makes sense. It's not something to be taken lightly. What about long-term effects? Are there any concerns about brain damage?
The research is still ongoing, but so far there's no evidence to suggest that responsible psychedelic use causes damage to the brain.
Okay, that's reassuring. I know there's been some talk about things like flashbacks or HPPD, hallucinogen persisting perception disorder.
Hmm.
What's the reality there?
Those are valid concerns. HPPD is a rare condition where people experience visual disturbances that linger after a psychedelic experience. We don't know exactly what causes it, but it seems to be more common in people who already have some mental health conditions.
So again, it comes back to responsible use, informed consent, and really understanding the potential risks. There's a lot of layers to this. It's not just about the science, it's also about ethics. Psychedelics have this long history of spiritual and ceremonial use. How do we balance that with scientific research?
That's an important point. We need to approach this research with cultural sensitivity and humility. Acknowledge and learn from indigenous cultures who have been working with these compounds for centuries.
So it's not just about isolating these molecules and studying them in a lab. There's a human element that we need to consider.
Absolutely. We need to have a respectful dialogue with these communities, make sure their voices are heard, and that they benefit from any research that comes out of it.
It's a complex landscape to navigate, but the potential benefits are really compelling. What other fascinating avenues is this research opening up?
Have you ever heard of a compound called huperzine A?
Huperzine A, can't say I have. What is it, and how does it connect to all of this?
It's a natural compound found in a type of Chinese club moss, and it's been shown to improve memory.
A memory booster from moss? That's incredible. How does it work?
So huperzine A inhibits an enzyme called acetylcholinesterase. That enzyme breaks down acetylcholine in the brain, and acetylcholine is a key neurotransmitter for learning and memory.
So by inhibiting that enzyme, huperzine A boosts acetylcholine levels. It's amazing that something found in nature can have such a specific effect on brain chemistry.
It really is. And because of its effects on memory, huperzine A is being studied for its potential to treat Alzheimer's disease.
So it could be like a natural alternative to some of the medications currently used for Alzheimer's. That's exciting.
It definitely shows promise. We need more research, of course. But it's a great example of how exploring the natural world can lead to some really important discoveries.
I'm starting to see a pattern here. It seems like there's this whole world of compounds out there, natural and synthetic, that can influence brain function in really profound ways. And we've barely scratched the surface.
You're right. The brain is incredibly complex, and we're still learning how to unlock its full potential. The discoveries we've been discussing, from DMT to huperzine A, are just a glimpse of what's possible.
It's both daunting and incredibly exciting. It feels like we're on the verge of a whole new era in neuroscience. What other tools are helping us unravel these mysteries?
Let's talk about toxins. Now, before you get alarmed, hear me out. Some of the deadliest toxins can actually be incredibly valuable for neuroscience research.
Toxins. Okay, that's not what I was expecting. How does that work?
Many toxins target very specific receptors or ion channels in the nervous system. By studying how they interact with the brain, scientists can get a much deeper understanding of how those receptors and channels function.
So it's like using a highly specialized key to unlock the secrets of a complex machine. But how do you study something that's potentially lethal?
Controlled environments and very careful research protocols. Take dendrotoxin, for example. It's a toxin found in the venom of the black mamba snake, one of the deadliest snakes in the world.
The black mamba. I've heard the stories. So how does something so dangerous become a research tool?
Dendrotoxin specifically blocks potassium channels in neurons, and these channels are essential for regulating the electrical activity of those neurons.
Right, they control the flow of information in the brain. What happens when you block them?
It disrupts the balance of electrical signaling, which can lead to paralysis and respiratory failure. Scary stuff. But understanding how dendrotoxin works is helping scientists figure out how potassium channels function and maybe even develop treatments for conditions that involve those channels.
I see. So by studying these toxins, we're learning about the inner workings of the brain, even though the toxins themselves are dangerous. What other toxins are being used this way?
Let's talk about bungarotoxin. It's from the venom of the krait snake, another highly venomous species. Bungarotoxin targets acetylcholine receptors.
Acetylcholine, right. That neurotransmitter involved in things like muscle movement and memory. So what happens when Bunger toxin messes with those receptors?
It binds to the receptors and blocks acetylcholine from binding, which can lead to paralysis. It basically disrupts the communication between nerves and muscles.
So it paralyzes the victim by interfering with the signals that control movement. How does this translate into research?
Because bungarotoxin is so specific to those acetylcholine receptors, it's been a really valuable tool for mapping those receptors in the brain.
Okay, I'm starting to get a sense of how these toxins, as dangerous as they are, can be really useful tools for understanding the brain.
It's a delicate balance, for sure, but the knowledge we gain can ultimately save lives. There's another toxin I think you'll find interesting, chlorotoxin.
Chlorotoxin, huh? What's the story of this one?
This one comes from the venom of the deathstalker scorpion.
Okay, the name alone is terrifying. What does it do?
It has this remarkable property. It targets glioma cells.
Glioma cells, though, the brain cancer cells, right?
That's right. And because it binds so specifically to glioma cells, scientists are looking at its potential for delivering anti-cancer drugs directly to those cells. That way, you minimize the damage to healthy brain tissue.
So it's like a guided missile zeroing in on the cancer cells.
Yeah.
It's incredible how nature provides these tools, even if they come from unexpected sources.
It really shows you the interconnectedness of the natural world and how even the most dangerous things can offer valuable insights. It's all about perspective.
This is definitely changing my perspective on toxins. But all this talk about specific receptors and targeted treatments makes me wonder about the bigger picture of how drugs work. Are we moving beyond the idea of one drug for one target?
That's a great observation. We are moving into the realm of polypharmacology.
Polypharmacology, that's a new one. What does that mean?
It's the idea that a drug can interact with multiple targets in the body, not just one specific one.
So it's like a master key that can unlock many doors. That seems like it would be a more powerful way to approach complex diseases.
Exactly. The traditional way has been to design drugs that target just one receptor or enzyme. But with polypharmacology, we can create drugs that address multiple pathways involved in a disease.
So it's a more holistic approach.
Yeah.
And I imagine it could be more effective. Are there any downsides to this approach?
Well, it gets complicated, of course. It requires a much deeper understanding of those interactions in the body, but the potential benefits are significant. By hitting multiple pathways, you could potentially get better outcomes and lower the risk of drug resistance.
Drug resistance is a huge problem with a lot of medications today. How does polypharmacology help with that?
Well, by hitting multiple targets, you're basically creating roadblocks for pathogens or cancer cells. It's much harder for them to develop resistance.
So it's like attacking the problem from all sides. Are there any real-world examples of this in action?
Absolutely. We're starting to see anti-cancer drugs that target multiple signaling pathways involved in tumor growth. And researchers are developing drugs for neurodegenerative diseases that target different pathways involved in cell death and inflammation.
It's amazing how quickly drug development is evolving.
Yeah.
It seems like all these new tools are opening up a whole new world of possibilities for treating complex diseases.
Yeah.
What other breakthroughs are helping us to understand how drugs work in the body?
That's where cryo-electron microscopy, or cryo-EM, comes in.
Cryo-EM. I have heard the term, but I'm not really sure what it is. How does it relate to drug development?
It's a revolutionary imaging technique that lets scientists visualize the structure of molecules in incredible detail, near atomic resolution.
Wow, near atomic resolution.
Yeah.
Are we talking about seeing the individual atoms that make up a molecule?
Almost. And this is really a game changer for drug development.
Okay, I'm intrigued. Tell me more.
Imagine being able to see the exact shape and structure of a protein that's involved in a disease. With that knowledge, scientists can design drugs that fit those proteins perfectly, like a key in a lock.
So it's like having a blueprint of the target that would allow for much more precise drug design.
Exactly. Cryo-EM is transforming how we understand how drugs interact with their targets, which is leading to much more effective and targeted therapies.
Okay, I'm starting to understand the significance of this technology, but how does it actually work?
It involves flash freezing samples of molecules in a very thin layer of ice. Then a beam of electrons is fired through the sample, and the way those electrons scatter is detected to create an image.
So it's like taking a super high-resolution snapshot of the molecule frozen in time.
That's a great way to think about it. And by combining thousands of those snapshots, scientists can create a 3D model of the molecule.
It's mind-blowing to think about the level of detail we can see now. It sounds like cryo-EM is a game changer, not just for drug development, but for our understanding of biology in general.
Absolutely. It's allowing us to see the molecular world in a way that we couldn't have imagined just a few decades ago.
Okay, I'm officially blown away by cryo-EM, but I have to admit, I'm a little overwhelmed by all this information.
It's a lot to take in, for sure, but I hope you're seeing the bigger picture here.
I am. It's clear that we're living in a time of incredible scientific discovery, especially when it comes to understanding the brain and the potential of things like psychedelics. It's a little daunting, but also very exciting.
I agree. We're on the cusp of a new era in neuroscience and pharmacology, one with so much potential for improving human health and well-being.
All right, I'm ready for whatever transformative factoid you've got up your sleeve next.
Then let's talk about gene editing.
Gene editing. That sounds like science fiction, but I have a feeling you're about to tell me it's closer to reality than I think.
You're right on the mark. And one of the most powerful tools in gene editing is CRISPR.
CRISPR, right. I've heard the term, but I don't really know what it is.
It stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's essentially a bacterial defense system that scientists have adapted to edit genes in all kinds of organisms, including humans.
So it's like a molecular scalpel that can cut and paste DNA.
That's a great analogy. CRISPR allows scientists to target specific genes and either disable them or make changes to their sequence.
The potential applications seem limitless. Are there any ethical concerns surrounding this technology?
There are definitely ethical questions, especially when it comes to editing the human genome. Things like unintended consequences, designer babies, and the potential for misuse.
So it's a technology that needs to be approached with a lot of caution and careful regulation. But the potential benefits are huge.
They are. CRISPR could potentially cure genetic diseases, engineer disease-resistant crops, and even create new cancer treatments.
It's clear that this is a powerful tool, one that could really change our world. But I'm curious about how it applies specifically to the brain. How is CRISPR being used in neuroscience research?
Well, it's opening up a whole new world of possibilities for studying brain development, function, and disease. For example, scientists can use CRISPR to create animal models of neurological disorders.
So they can recreate the genetic mutations that cause these disorders. That seems like it would be really valuable for understanding how they develop.
Exactly. By studying these models, scientists can gain insight into potential treatment targets.
What about using CRISPR to treat brain disorders in humans directly? Is that a possibility?
It's still very early, but researchers are exploring the possibility of using CRISPR to correct genetic defects that cause neurodegenerative diseases, like Huntington's disease.
So it's like editing out the faulty genes that lead to these devastating diseases.
That's the idea. But delivering CRISPR to the brain is a big challenge. Scientists are trying to develop safe and effective ways to get the CRISPR machinery into the right cells.
It sounds like a complex process. But if those challenges can be overcome, the potential is incredible.
It really is. If we can harness the power of CRISPR, we could potentially cure diseases that we once thought were incurable.
This deep dive has been full of amazing revelations. We've gone from exploring the mysteries of consciousness to the frontiers of genetic engineering, feeling a little overwhelmed, but also really inspired.
That's great to hear. We've covered a lot, but I hope you've gained a new appreciation for the incredible complexity and wonder of the brain.
I have, and I'm excited to keep learning more. Are you ready for the final leg of our journey?
I am. Let's continue our exploration with a discussion of nootropics.
Nootropics, those are the smart drugs everyone's talking about, right?
That's right. They're substances that are thought to boost cognitive function, memory, creativity, or motivation.
Brain boosters. Interesting. But do they actually work?
The evidence is kind of mixed. Some nootropics, like caffeine, have been studied really extensively, and their effects are well documented.
Oh, I can definitely vouch for caffeine. I wouldn't be able to function without it.
But for other nootropics, the evidence isn't so clear-cut. There are some promising studies, but we need more research to really understand how they work and what their long-term effects might be.
So it's important to be cautious and do your homework before trying anything new. But I'm curious about the potential here. If they live up to the hype, could they really change the way we learn, work, and even age?
Those are the questions scientists and ethicists are wrestling with right now. The idea of enhancing our cognitive abilities is both exciting and a bit unnerving.
I can see why. It makes you think about fairness, accessibility, and even what it means to be human.
Exactly. If we could enhance our brains with a pill, would that change how we value things like hard work and natural ability?
It's almost like we'd be moving into this realm of transhumanism, using technology to augment our own biology. Where do we draw the line?
That's the conversation we need to be having as a society. But I understand your intrigue. It's a fascinating thought to imagine unlocking the full potential of our brains with the help of these substances. It could lead to breakthroughs in all sorts of fields: science, art, technology.
It's definitely a tantalizing possibility.
But for now, we need more research to understand both the risks and the rewards.
Wow, we've covered so much ground already, from ego resets and DMT to toxins and gene editing. It's mind-blowing.
It really highlights just how diverse and interconnected the world of brain science is. We've touched on so many different approaches to understanding and manipulating how the brain works.
And you're telling me there's still more?
Oh, there's always more to discover. Let's talk about a field that's really pushing the boundaries of what we think is possible: brain-computer interfaces.
Brain-computer interfaces. That sounds straight out of science fiction. Or are you talking about plugging our brains into computers?
It's not as far-fetched as you might think. Brain-computer interfaces, or BCIs, allow for direct communication between the brain and an external device.
So it's like creating a bridge between our thoughts and technology. What does that actually look like in practice?
Well, imagine a paralyzed patient being able to control a robotic arm just by thinking about it, or someone with locked-in syndrome being able to communicate using a computer.
That's incredible. We're talking about restoring function, giving people back their independence. How does that even work?
There are different types of BCIs, but a common approach is to implant electrodes in the brain to record the activity of neurons. Those signals are then decoded and translated into commands that control a device.
So you're basically reading the electrical signals from neurons and turning them into instructions for a machine. That's wild. What are the limitations of this technology right now?
Well, it's a relatively new field, so there are definitely some technical hurdles. The electrodes need to be biocompatible. They can't cause any harm to the brain. And we need to develop better algorithms to really accurately decode brain signals.
I can imagine. But even with those challenges, the potential here is enormous. We're not just talking about restoring lost function. Could we potentially enhance existing capabilities?
That's the really mind-boggling part. Imagine being able to control your environment with just your thoughts, or being able to access information directly from the internet. The possibilities are pretty incredible.
It's exciting, but also kind of scary. Are there any ethical concerns about this technology?
Absolutely. There are all sorts of ethical issues we need to think about very carefully. Things like privacy, autonomy, and even personal identity. What does it mean to be human when we start merging our minds with machines?
It's like we're entering a whole new phase of human evolution, one where technology is blurring the lines between the physical and the digital.
It's a profound shift, and one that we need to approach with careful thought and planning.
This deep dive has been such an incredible journey. We've explored everything from the mysteries of consciousness to the potential of brain-computer interfaces. I'm feeling a sense of awe and just incredible respect for how complex the human brain really is.
That's wonderful to hear, and I hope this sparks a desire to continue exploring this field. The more we learn about the brain, the more we realize how much more there is to learn.
Thanks for guiding us through this amazing landscape of brain science. I can't wait for our next deep dive together.
Until then, keep those minds curious and open to all the incredible possibilities the human brain has to offer.